Hello friends, let us discuss active side of enzymes. Enzymes bind to substrate to produce enzyme-substrate complex. Most enzymes are proteins and they are made up of multiple amino acid residues. I am not using the word all. I'm using most instead of that. All enzymes are not proteins. Why? Because of ribozymes. Ribozymes are catalytic RNAs. These are non-protein enzymes. Can you name a few ribozymes? snRNA, which is involved in splicing. 28S ribosomal RNA, which has peptidyl transferase activity. 28S rRNA is a component of ribosome in eukaryotes. There are certain self-splicing introns. So these introns, they splice themselves. There is no need of snRNA and spliceosomes. There is one RNase P. This is involved in trimming of transfer RNA during the post-transcriptional modification. This is also a ribozyme. At least try to remember the first three: snRNA, peptidyl transferase of ribosome, and self-splicing introns are ribozymes. Right. So let's go back to our previous slide. Most enzymes are proteins and they are made up of amino acid residues. The entire protein portion of enzymes is not involved in catalysis. Catalysis takes place only in a cleft or crevice of the enzyme. Then what is the need for the rest of the protein portion? Can you think of something? Rest of the portion acts as a scaffold. It provides stability. Also, it may have regulatory function. There are two components of active site. What are they? The first one is substrate binding site and the other one is catalytic site. This is very, very important. Most people think that active site means it is catalytic site. No, there are two separate sites in the active site. Substrate binding site will also bind to the coenzymes. Many times, coenzymes are considered as co-substrates in certain chemical reactions. Okay. So substrate binding site and coenzyme binding site is separate from the catalytic site. Remember this. Active site may contain certain motifs. First of all, we should know what are motifs. Motifs are conserved sequences of amino acids in proteins that form a distinctive structural or functional unit. Let us discuss few important motifs in enzymes. These motifs may have role in catalysis, ligand binding and protein-protein interactions. We will discuss just two motifs, Rao-Roseman fold and Zinc-finger motif. Look at the structure of Rao-Roseman fold. What are you seeing? Look at this. What is this arrow indicating? This is indicating beta plated sheet, right? And look at the direction of the arrows. So they are all in same direction. That means this is a parallel beta sheet, right? And this beta sheet is flanked by alpha helices. So Roseman fold is NAD binding motif consisting of beta sheet flanked by alpha helices. There is no need to memorize this beta sheet, alpha helices and all. Just for your understanding, I am telling you. What you have to remember is Roseman fold is NAD binding motif. This is all. This is all you need to Remember, okay? So it binds to NAD, nicotinamide adenine dinucleotide. So what is this NAD, FMN, FAD and all? Do you want to know more? So here is the link. So you can click this link and watch the video. Rao-Roseman fold is present in certain dehydrogenases which utilize NAD to accept electron. They are alcohol dehydrogenase and lactate dehydrogenase. There are many other dehydrogenase. Just remember a few. The next motif we are going to study is Zinc-finger motif. Look at the diagram. What are you seeing? You are seeing zinc and you are seeing certain amino acids. So look at this. There is yellow color. Can you recognize this yellow color? Let me zoom it. See this. See this yellow color, right? So yellow usually used to indicate sulfur. That means that amino acid is a sulfur containing amino acid. So this is cysteine. Okay. This ring, again, let me zoom. Can you recognize what kind of ring is this? This is imidazole ring. Do you remember the imidazole ring containing amino acid? That is histidine. The zinc finger motif consists of one or more zinc ions coordinated by cysteine that contains sulfur and histidine residues. Zinc finger motif is involved in DNA or RNA binding. Zinc finger motif containing proteins are involved in regulation of gene expression. There are certain enzymes that also contain zinc finger motif. So you should never forget zinc finger motif. Zinc finger motif binds to DNA. Remember this, very, very important. Zinc finger motif is a DNA binding motif. All right. So now we can discuss the properties of active site. In the exam, if they ask you what are the properties of active site, just write what I am going to discuss. Active site is a three-dimensional crevice or cavity created by amino acid residues originating from various regions of protein's primary structure. What do I mean by this? See, in the primary structure, what is primary structure? It is the linear sequence of amino acids from amino terminal to carboxy terminal. One amino acid can be here, the other amino acid can be here, the other can be here. During protein folding, these three amino acids may come closer to each other. So that is the three-dimensional structure, right? So active site is a three-dimensional structure. Amino acids which are far away in their primary structure can come closer in the tertiary structure to form active site. That is what I am trying to explain. I hope you have understood this. Substrates are bound to enzymes in active site by multiple weak non-covalent interactions. Very, very important. The enzyme-substrate interaction is usually non-covalent interactions. What are those non-covalent interactions? Electrostatic interaction, hydrogen bonding, van der Waals forces and hydrophobic interactions. The active site is complementary to the transition state rather than the substrate. What do I mean by transition state? Have you ever heard of this term, transition state? Let me explain this. Look at this. In the vertical axis, free energy is plotted. G stands for Gibbs free energy. In the horizontal axis, it is the reaction coordinate. Reaction coordinate explains the position of substrate and product. So here is your substrate, here is your product. Compare the free energy of substrate and product. See, the free energy of product is lower than the free energy of substrate. What is delta G? Delta G is the difference between free energy of product and free energy of substrate. So delta G here, it will be negative, less than 0. That means this is an exergonic reaction. What is delta G? The difference between the free energy of product and the free energy of substrate. If delta G is negative, the reaction is possible spontaneously. Thermodynamically, the reaction is possible. Alright. Whether the reaction is endergonic or exergonic, substrate always needs to be activated to transition state. So we use the symbol double dagger to denote the transition state. So this is the transition state. Conversion of substrate to transition state is always endergonic reaction. The energy that is needed to convert substrate to transition state is activation energy. Activation energy is denoted by delta G double dagger. Okay, this is like a dagger, right? So that is why double dagger. Remember, delta G double dagger is your activation energy. Enzymes, they decrease the activation energy. Okay, so in enzyme catalyzed reaction, what is going to happen? They will take a different route and they will decrease the activation energy. So this is without enzymes and this is with enzymes. How does enzymes decrease the activation energy? It is because of binding of substrate to enzymes release some energy. That energy is known as binding energy. So this binding energy is used to decrease the activation energy. Let's not go too much deep into this thermodynamics. So active site is complementary to the transition state rather than substrate. Another important property of active site is that water is excluded from the active site until unless it is a reactant. This is known as desolvation of active site. I will give you an excellent example of desolvation of active site later. Remember this word. We have already explained this in this video. Click this link. You can watch about the substrate specificity of serine proteases in this video. The interaction between active site and the substrate can be explained by two models. The one is lock and key, the other is induced fit model. Emil Fischer, he assumed that the active site of enzyme is similar to a lock and the substrate is the key. So according to his theory, there needs an exact fit between lock and the key. That is exact fit between the substrate and enzyme. Look at the image. See this. This is a rigid portion, active site, and this is the key. So that's what he assumed. But lock and key model has been proven to be wrong. So what is the most accepted model? That is induced fit model of Daniel Koshland. So look at this image. So this is your active site and this is your substrate. Binding of substrate, it induces conformational change in the enzyme. Let me give you an example for induced fit model. Also for this desolvation of active site. Remember what is desolvation of active site? Until unless water is a reactant, it is always excluded from the active site. What enzyme is this? This is nothing but hexokinase, the first enzyme of glycolysis. What are the substrates? Glucose plus ATP. Glucose plus ATP. Glucose will be converted to glucose 6-phosphate. Right? To the OH group, phosphoryl group is added. That is the reaction. So in this reaction, once the substrate binds, look at this. The active site is closed. See this. So water is excluded from the active site. You all know that the whole biochemical reactions are happening in the milieu of water, aqueous environment. Why do we need to exclude water? It is because, see, in glucose also you have OH group. In water also you have OH group. So what can happen? Instead of OH group of glucose, phosphoryl group can be added to the OH group of water. To avoid this, to avoid wastage of ATP, nature has gone for this induced fit mechanism to exclude water from the active site until unless it is a reactant. For hydrolase enzymes, water is the substrate. So in hydrolase enzymes, water is not excluded. Alright? Okay. So we are going to complete this lesson with these three important terminologies. In the exam, if they ask you about properties of active site, just write whatever I have mentioned. So this is something extra which I am going to teach you. What are the important terminologies? We are going to learn bifunctional enzymes, multifunctional enzymes, and multi-enzyme complexes. So this is based on active site only. That is why we are learning this in active site lesson. Bifunctional enzyme is a type of multifunctional enzyme. So what are multifunctional enzymes? Usually, one enzyme will have only one active site. There will be only one active site. There are certain enzymes which can have more than one active sites. Active site 1, active site 2. If there are two active sites in a single protein, that is known as bifunctional enzymes. Bi means two. So it has two active sites. So that means it will have two functions, bifunctional enzymes. Clear? So this is a single protein. Remember, these are not two different proteins. This is a single protein. In the same protein, there are two active sites. Let me give you examples. Bisphosphoglycerate mutase. Where have we studied this enzyme? In a different video, we have already learned this. 2,3-BPG shunt. You remember? Rappaport-Lubering shunt. Click this link. You can watch Rappaport-Lubering shunt. Phosphofructokinase 2 is involved in the production of fructose 2,6-bisphosphate, which is a potent allosteric activator of the rate limiting step of glycolysis, that is PFK1. Fructose 2,6-bisphosphate, it activates PFK1. So fructose 2,6-bisphosphate is produced by PFK2. So this enzyme, it has two activities. One is kinase activity and the other is phosphatase activity. So that is why this is a bifunctional enzyme. In glycogen metabolism, we might have studied this enzyme, that is debranching enzyme. This has glucan transferase activity as well as glucosidase activity. So there are two activities in this. What about UMP synthase? So this enzyme is involved in pyrimidine biosynthesis. So this has two activities. It's going to add phosphoribosyl pyrophosphate to orotic acid. Orotic acid plus PRPP. It produces OMP. That is the first activity. So that is orotate phosphoribosyl transferase activity. The next activity is decarboxylation of OMP to produce UMP. So that activity is very easy. OMP decarboxylase activity, right? So phosphoribosyl transferase activity and decarboxylase activity. So these are the two activities. Defect in UMP synthase leads to orotic aciduria. So there is a separate video on orotic aciduria, which you can watch later. Let us differentiate multifunctional enzymes and multi-enzyme complexes. What are multifunctional enzymes? As I told you, bifunctional enzymes are a type of multifunctional enzymes. Some enzymes, they can have more than 5 or 6, even 6 enzyme activities in a single polypeptide. For example, fatty acid synthase. Fatty acid synthase, it has multiple enzyme activities in a single polypeptide chain. So it is a multifunctional enzymes. What are multi-enzyme complex? These are different enzymes, right? These are 3 different enzymes. For example, pyruvate dehydrogenase complex, alpha ketoglutarate dehydrogenase complex, branched chain keto acid dehydrogenase complex. So all these complexes are made up of 3 different enzymes which are linked. Okay, so 3 different enzymes are assembled together to form a multi-enzyme complexes. Whereas multifunctional enzyme is a single polypeptide chain, single protein. Multi-enzyme complexes are different proteins, right? In multifunctional enzymes, there is something called substrate channeling. That means in fatty acid synthase, if the substrate comes, ultimately it will leave the enzyme as a product only. The intermediates of the reactions are passed between the active sites without being released into the solvent. So this increases the efficiency of the enzyme. Whereas there is no substrate channeling in the multi-enzyme complex. Pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase, and branched chain keto acid dehydrogenase, BCKD. So these are the few examples for multi-enzyme complexes. So with this, I think we can finish active site of enzymes lesson. If you think this video is worth watching, please share it with your friends. Thank you.